next presentation is on integrating metabolomic data into metabolic models. And so this is an exciting follow-up to yesterday's discussion, which was all about metabolomics data. And so we're excited to hear this from Chris. And so Chris, go ahead and take it away.
All right, I'm going to share my screen. Can you guys see that?
Yeah, I see your Discord screen, and there's your slides.
And I can't, my button is hiding from me. There we go. All right, can you see the slides?
Yep, now I see your slide in presentation mode.
All right.
Great.
So this will be a tutorial on the integration. Oh, why is that? Okay, I'll just click. There you go. It's a tutorial on the integration of metabolomics and the modeling. And the day 2 workflow that you saw yesterday really covers this. FTICR metabolomics to M over Z values for peaks. And then predicted formula using the formulator tool. And then a whole bunch of analysis you can do with the output of that. So that's not really tying in the genomics data yet. So it's focused on the metabolomics side, although some of the statistical analysis does let you correlate to that data. So predicted property analysis is predicting things like thermodynamics and other properties based on formula aromaticity, things like that. Chemical transformation prediction, which can be done both from mass differences between peaks or formula. Transformation network analysis, so just making a network out of these predicted formulations, statistical analysis, correlating peaks with various things, and then the lambda analysis that he uncovered. And in day 1, you have been through metagenomics. Kelly and the other presenters cover how you convert metagenomes and the contigs to either a annotated metagenome assembly, with which Jonica, I think, just talked about, and then bins or binning, and then annotation of bins into what we call metagenome assembled genomes, or MAGs. And then what Jonica just covered is the metagenome models we can build from the annotated metagenome assembly, which is sort of a giant mixed bag of your microbiome, or these genome models that can be built from bins, and then the gap filling and flux balance analysis. But how to tie these two worlds together? And the answer is, or one of the answers is, cheminformatics. So we want to link models to metabolomics data, and the step 1 for doing that is to map the metabolomics peaks to the model. So we can use a formula match. We generally don't do this with mass. At the very least, we want to have a formula from Formulator. Now remember yesterday, Formulator is not going to give you a formula for every mass. But it'll give you a formula for a lot of them, typically about 5,000 for these FTICR data sets that I've seen. And this is weak, and the reason is because many different compounds, potentially very different compounds, have identical formula. Or a structure match, which is much better, but still not perfect because of stereoisomers. So what I mean by structure match is something like a SMILE string. If they give you an Enchi key, and they will, if you do MSMS with Trent Northern or Oliver Fien or one of these metabolomics labs, or at Enzel with Tom, if they give you an Enchi key, just be aware, just go ahead and strip the stereoisomer information off of that Enchi key and do a match on the base of that key, because metabolomics can't give you stereoisomerization at the moment. I mean, there are some exceptions, and talk to Josh, and Josh Adkins, he can tell you about what they can do with the cutting edge stuff today, but generally expect that you'll get a non-stereoisomer base structure. A structure map, though, is much better. There's far fewer stereoisomer redundancy in the chemical networks than there is, for example, formula redundancy. And then, currently mass spec is, yeah, that's already made that point. So there's 4 scenarios that then arise when you try to do this mapping. The first scenario 1 is you know what the metabolite is, you know what the structure is. Again, FTICR, you're generally not going to get a structure. It's not MSMS. They're working on that, but currently it doesn't do that. But if they do have the structure from it, if you run MSMS, then you know what the structure is. You find it in the model. That's scenario 1. Great, it's in your model. You can run FBA, you can explore how the model uses that metabolite, you can model the metabolomics data. Scenario 2, the metabolites in the database, you find it in the database, it's a structure match, we know what it is, but it's not in our model. Well, Jonica just showed you the gap filling tools. We can make the model make that metabolite. And so we can integrate the metabolite into the model that way, and now we have this additional evidence-based way of filling holes in the model, which is very cool. It's what we want to do. And once we've got it in the model using the gap filling, then we can go to this, running FBA to study the metabolite. Scenario 3, we have the structure. We know what the metabolite is structurally, but we do not know what pathways to, it's not in the databases. The compound is not in the databases. So we have no pathway to the structure. And this is where the cheminformatics comes in. But also scenario 4, which unfortunately with FTICR, this is where you're at the moment. You have a formula, you don't actually have a structure. So it's sort of this hypothetical thing. But again, the cheminformatics offers a mechanism to get at those formula with a mechanistic basis. So what you saw in day 2 was sort of non-mechanistic formula-derived analysis. And there's a lot of very powerful things you can do with that. What we're going to talk about here is, okay, can we mechanistically do anything to mechanistically understand why those formula are there? And again, this is where your FTICR data is formula scenario 4. So overarching challenge with linking metabolomics to models is that there's this dark matter problem. People talk, you've heard dark matter in biology before, and it's usually referring to proteins of unknown function, but there's dark matter metabolomics too. There's metabolites of unknown, that we don't think that we see in biological systems, but are not in biological databases. There's no universal method for measuring small molecules, so that's a contributing factor to this challenge, is that there's just a lot of different, I talked about FTICR versus LCMS, MSMS, and FTICR can be LCMS if you put some separations in front of it. So generally, it's only possible to confidently identify 5 to 20% of the features in an MSMS data set. So if you don't have MSMS, you're not confidently identifying anything. You can get a molecular formula with fair level of confidence for a large fraction of the peaks with FTICR. That's one of the advantages of that method, is the highly fine resolution on the masses gives you a pretty good idea of what the formula is. But even if you have MSMS, you only get 5 to 20%. Why? Because the other 80% aren't in structured databases, and you can't. MSMS identification relies on knowing a structure, predicting how it will decompose, and then comparing those predictions to your MSMS fragmentation pattern. So there's this other issue that if the structure isn't in the database, you're going to have a hard time finding the compound using this technology. Or what they really do with MSMS is acquire a sample of the compound and run it side by side. That's called running a standard in metabolomics. And so again, can't get a standard for this complete, dark matter molecule nobody's seen in a system before. But it leads to this, cycle of the models are incomplete. So because the models and the knowledge is incomplete, we can't, properly annotate our data, which leads to incomplete knowledge. So this is Kelly talked about this same problem in annotation. The protein function knowledge is incomplete. So if our knowledge is incomplete, then we can't annotate completely. And because we can't annotate completely, we lead to, incomplete knowledge. So we have this same cycle in metabolomics data. Well, we have a way, cheminformatics offers one mechanism to get around this. But remember, you're at this point, as soon as you start doing cheminformatics, just to be clear, we're diving down the rabbit hole. We're going to Wonderland. This is, computational prediction of potential novel reactions. So it's not real until you prove that it's real, is what I would say. So we have these generalized reaction rules that are learned from biochemistry databases, and we can apply these rules systematically to a set of compounds that we're interested in and predict reactions and compounds that could arise, new reactions and new compounds, which sort of addresses this missing knowledge problem. And then those new structures that are predicted could give us predicted MS and allow us to address this other 80%. And then we search for evidence of these pathways and compounds in our metabolomics and genomics data. So we have three possible hypotheses of what could lead to these novel reactions. We have generalist enzymes. We've got an enzyme that it does a mechanism, it does a chemical activity that has been observed before, like oxidation and reduction with NAD and NADH. But nobody's seen an enzyme operate on this specific substrate. So it's doing a known activity on a new substrate. So we call that promiscuity, although in many cases this is not promiscuity. This is an enzyme, this is its primary activity. It's just the primary activity of that enzyme is something we've never seen before. And so that enzyme is either unannotated or incorrectly annotated. So be aware of that. When you have an annotation of an enzyme, it might do that activity, but it might also do something similar to that activity. You have an unannotated enzyme, so it could just be chemical mechanisms that we have not discovered yet, and there's going to be a lot of those. Or spontaneous reactions. So don't forget, this is chemistry. Chemistry happens whether biology is there to make it happen faster or not. So always be aware of this kind of hidden, unpredictable monster in your system. Chemistry is going to happen no matter what, particularly if you have a higher temperature system or extreme pH, or oxygen, anything that in the environment that contributes to faster, just natural chemistry. So how do we use cheminformatics to get at these? So I'm going to talk about this one, the generalist enzymes and the spontaneous reactions. This is like really far out there, computational chemistry. There's people thinking about this, but it's still less advanced. So we build enzymatic rules to predict new reactions based on known mechanisms. And here's an example of how we do this. So if we look at these 4 different EC numbers with 4 different reactions, but we actually look at the chemistry that's happening, we see that they all follow the same pattern. There's this functional group, water's being added, it's being hydrated. You have to have these keto groups there. If you do not, this chemistry doesn't happen. And then this bond can be broken and form a carboxylic acid and a terminal alkane, a methyl group. So we've learned these. This is based originally on work with Hatzmanakatas et al. Actually stuff I did in my PhD. And then we have a set of 210 rules learned from this, and Vasily Hatzmanakatas has gone on, and others have gone on to learn many, many more rules. So this is, there are many labs working on this, building better and better rules learned from these databases, and I think machine learning is going to blow this wide open. For spontaneous chemistry, we've also developed rules, and this is work my lab does in collaboration with Andrew Hansen and Oliver Fien. But we have 150 spontaneous rules learned from spontaneous reactions in various databases. So we look at those spontaneous reactions and we develop generalized rules that represent with that the chemistry that happens with those spontaneous reactions. So we have rule sets for both spontaneous and enzymatic rules. To discover new pathways, we can apply these rules to a set of starting compounds. So we take all of our rules and apply them to a set of starting compounds. And we get new compounds and new reactions. So this is the novel reaction prediction and novel compound prediction. We can apply them once, but we could just go ahead and apply them again and again and again and get whole long pathways. And the observant among you will quickly say, but Chris, you're going to have about 1 billion compounds after 4 or 5 generations, and you're absolutely right. And so we have to have ways of dealing with that. And I'll talk about that here. Of course, for what we call mines, which are metabolic and silical network expansions, we can also just look at applying our rules to all all known biochemical compounds for one generation and make this giant hypothetical database to try and deal with this dark matter problem. And we've done this with KEGG, EchoPsych, and YMDB, and this is what you get. So you have about 15 to 20,000 compounds in KEGG. Initially, we apply our rules. We have almost 1 million compounds and reactions in the mine, which is just an expansion of KEGG with our enzymatic rules. And we've done this with our spontaneous rules recently as well. And we've done this with EchoPsych and YMDB, which is a yeast database, and EchoPsych is E. coli. And we get about a 50x expansion in number of compounds. And there's a site you can go to called minedatabase.mcs.anl.gov to explore that stuff. So take a look if you want to see what, at this site, if you want to explore the chemistry that gets produced. But that's one iteration of our rules. So if your pathway is 2 or 3 steps out from the known knowledge, the mine isn't going to help you. Now, one of the nice properties of this database, if you compare the contents of the mine to something like PubChem, which of course also has more than 1 million compounds. It has tens of millions, I think, at this point. But PubChem is a general database, not a bio database. And you can see that here. If you look at natural product likeness of the PubChem database, the mine expansion of KEGG looks a lot more like KEGG than it does PubChem. So that's a nice, it's more biological. What does the mine and cheminformatics let you do with metabolomics data? Well, for example, I've got this compound that was observed with metabolomics data, based on MSMS, we know that it's S-succinyl glutathione, but it wasn't in the database. Well, the cheminformatics predicted that, well, if you combine glutathione and fumarate in this novel reaction, it'll make this compound. And here's the evidence for that reaction to happen. Well, cysteine will combine with fumarate and form, succinyl cysteine. And so you can say, well, then if this can happen, then maybe this can happen as well. And so I've now mechanistically come up with a hypothesis of how this peak came to be. And I can look for enzymes now that might do this, isolate the enzyme, purify it, test it in a test tube, and see if the chemistry happens. Same thing with this second reaction. So this has been successfully applied for a couple of compounds in E. coli. So look, we actually came up with new compounds and the most studied bacteria ever. And we still found new stuff. There's C. reinhardtii and human cancer cells. So there's new chemistry happening in human cancer cells because the cells are all out of whack. And then Artemisia douglasiana. So there's a number of new discoveries that come from this cheminformatic expansion. But what do we want to do? We want mechanism and we want enzymes, not just hypothetical reactions. So how do we find the genes once we have a reaction that can make the metabolite that we see in the FTICR data? How do we find the gene that might be responsible for making that? Well, if you go back to what Kelly was talking about on Monday, you have annotation, which has this just fundamental strategy that if I want to know the gene that does this reaction in organism A, I can go to a database of other organisms and find genes that do the reaction there, and just do a, look for homology and then say, oh, well, gene 43 in organism A is homologous to these, highly homologous, and so I think it's the one doing this reaction. That is the fundamental basis for most genome annotation that happens today. But what about cheminformatics? Well, reaction 2 is a novel reaction. I go to my database, I am out of luck. There is no reaction 2 in my database. There are no genes. So what do I do instead? Well, it goes back to what I described earlier. If you have the, let me get this bumped out of the way. If you know similar reactions, you can find similar known reactions in the database that maybe have the same chemical mechanism, maybe the same cofactors, and a very similar primary substrate. Then you can say, well, the genes that do this probably with some mutation or modification could do this as well. And so I'm going to argue like, well, let's do a reverse lookup on a BLAST and see if, okay, this gene, come up with candidates to do this. And you'll have to go with lower similarity thresholds if you do this. And you're going to get a lot of candidates. So the one thing, we have successfully applied this to find genes that for novel reactions that then we successfully tested in the lab. We're not the only ones, others have done this. So this does work, but you get a lot of candidates. You generally want to combine it with other sources of evidence. Like in our case, we did transcriptomics. We were interested in a pathway that degrades a specific compound of interest. So we used our cheminformatics to predict the pathway. We applied this approach to predict genes for the pathway. But then we also did RNA-seq on cells growing on that compound and another compound, and we looked at differential expression. So crossing differential expression with genes predicted through this approach gave us on the order of 10 candidates. That is a very reasonable number of candidates to test in the lab, and 2 of those turned out to have the activity. So that's kind of the workflow here. So, okay, I've gotten you, hopefully excited about cheminformatics, and you're like, well, okay, I want to try this. How do I do that? So we have an app in KBase that lets you run cheminformatics. The app is called Pickaxe. Pickaxe is not an acronym. I generally don't like to use acronyms for my tools because I think you end up with names that have nothing to do with what the tool does. So I chose Pickaxe, which also has nothing to do with what my tool does. But it's because Pickaxe is a tool for creating mines, which ironically enough is an acronym. And the mines are, again, metabolic and silico network expansions. So the Pickaxe runs the cheminformatics tools on a set of input compounds and produces predicted novel reactions and compounds. It's based on an open source toolkit called RDKit. RDKit is amazing. Thank God they made it open source. It basically, if you read anybody doing cheminformatics and novel reaction prediction, I would bet you even money that they're using RDKit, and I would win most of the time. So it includes, Pickaxe has tools for visualization and rapid creation of reaction rules in our MINE website. It's there. Multiprocessing, database, and chirality. All that's built into the MINE database. And it uses smarts to describe the reaction rules. And that's because RDKit uses smarts. Smart strings look like this. Basically, it's the dot stands for 2 different substrates. So there's 2 different reactive sites. And basically it says look for this smart string in one molecule, look for this smart string in another molecule, and convert those structures that correspond to these smart strings into this, and you'll have your product. And that's how it works. So, this smart string corresponds to this structure, this corresponds to this structure, and the rule when applied makes this structure. So how does Pickaxe work? So you start by specifying your starting compounds, which can be a model, an entire model, so it'll use every compound in the model in that case, or a compound set, which you can import from a TSV in KBase. And so it could be like one compound in a compound set, or multiple compounds. And you specify rule sets you're interested in, and I'll show you what the UI looks like in a minute. Which could be, right now is limited to our enzymatic or spontaneous rules. If you're out there making awesome cheminformatics rules, please email me. I would love to have your rules in the system. We're working on adding retro rules, but there's some technical challenges to doing that. But we also collaborate with the Broadbelt and Tayo lab, and they're working on some really nice rules too. You can specify the number of generations you want to apply. Here I'm running 2 generations, which means I run my rules once, I get generation 1. I run my rules again, I get generation 2. I actually wouldn't get these return reactions until I run my rules a 3rd time. So that's just, you get the return reactions once you run on the current generation. But that does happen. You will get predicted reactions that go the opposite way. Rules are applied iteratively, but we talked earlier about combinatorial explosion. Yes, I hear you. It happens. It's a very big problem. So we have to apply filters, and the way we can limit the compounds retained in each generation, that's how Pickaxe works. You give it a limit. You say, I know you made 50,000 compounds. I only want to keep 3,000 of them. And so how does it pick which 3,000 to keep? Well, you can say, well, I'll keep everything that hits the known databases, the model seed, which includes KEGG and Metapsych. So model seed is a merger of KEGG and Metapsych. We have a paper coming out on model seed and NAR this year. So hopefully. It's in revision. And then we have, you can specify a target list of compounds that you absolutely want to keep if you encounter them. So if you're looking for pathways to a very specific molecule, you can say like, I want to keep that if we hit it. And this is where the crucial thing for us comes in. You can say, I want to keep any compound that hits a peak in my metabolomics data. And then the filtering will strip out things that don't, randomly strip out things that don't meet these criteria until it gets down to your limit per generation. So here's what the UI looks like in KBase. You guys are pretty familiar with KBase apps at this point. So here's where you input your starting compounds. Again, you can give it an entire model or a compound set. Here's where you give your target compounds. That's a compound set only. Here's where you get your metabolomics status. You give it this chemical abundance matrix. And you can specify, you can choose not to specify a condition if you want, and it will just try to hit any metabolite that's mentioned in this matrix, or you can specify a condition here. And then it will only choose remember your conditions are presence, absence. So the way it chooses is if there's a non-zero value, it's going to assume it's there and choose it. So if you have presence, absence data, this will work perfectly, but there's no thresholding mechanism in here at this point. Then you pick a rule set. You can pick multiple rule sets. You can run the spontaneous and enzymatic rules at once if you'd want to. You give a number of generations. The default is 1, so if you want multiple generations, you'll have to adjust that. And then the maximum hits per peak. So here's the thing. Once you hit a peak with a compound, and it's going to do formula matching for the FTICR data. It is capable, although this isn't tested, so if it doesn't work well, don't come after me yet. It is hypothetically capable of handling cheminformatics data that's annotated with Enchi strings or smiles as well, not just formula. But you get hits per peak. One problem is once it finds a single compound that matches the formula in a peak, it is very good at then finding more compounds that match the formula in that peak. And so it will rapidly find thousands of small variations, like I moved this double bond here and look, I found another one. Lots of small variations of compounds that can hit a peak. And so we establish a limit, and it will guarantee you to keep the first 10. It doesn't mean it won't keep beyond the first 10, but it will guarantee you the first 10 if you pick 10. And then max compounds per generation, you pick 3,000. So this is all about being able to explore farther and down the rabbit hole and not run into the combinatorial explosion. That's the whole idea of the filtering mechanism. Currently, the filtering is random, so if it makes 100,000 compounds, it's going to randomly discard 97,000 until it gets to 3,000. Eventually, I'd like to implement structural diversity as the criteria instead, but that's not happening yet. And you can use these criteria if you're like, I don't care about making model seed compounds. You can uncheck that box. And finally, you give the name. So the output of this thing is a model. The nice thing about that, if you're like, well, I want to use these pathways for this, that, and the other thing that KBase doesn't do, but I know Cobra Pi or Cobra Toolbox does do, you can export that model with all the chemistry predicted by this tool into SBML, or you can export as a TSV or whatever format you want. Or in the gap filling tool, you have the ability of specifying this model as a additional database for gap filling. So it will then use this chemistry to gap fill your model. So if you're like, I want to grow my model on, this compound, and there's no pathway for consuming that compound, well, I'm going to run cheminformatics on that compound first, and then attempt to gap fill my model on the cheminformatics predicted chemistry plus the standard biochemistry from the model seed, and it will combine both into a single network and gap fill from it. So that's very powerful. Basically lets you fully integrate a metabolic model with hypothetical chemistry, with metabolomics data, which is what we want to do here. So that's the acknowledgements. I know I've got some time left. I'm going to jump into, I want to talk about the acknowledgements are important, so I'll just quickly This is the KBase team, and this is some subset of my group that managed to get together for a photo. We do have beaches in Chicago. I know you guys all think we're a frozen wasteland, but we have beaches. You can't go to them now, but someday you can go to those beaches. So I want to show you what the output of Pickaxe looks like. I'm going to jump into this narrative. This is the output. It gives you an overview. So it says, in this case, I ran for 3 generations on I believe a metagenome model. And it gave me this output. And I'll start with the overview. So it tells you the number of rules it ran. I ran with both our enzymatic rules and our spontaneous rules. So 344 of them. There were more than 344 rules run. 344 produced a reaction. So that's what that number is. This is the number of reactions and compounds produced after filtering. So the filter has been applied. This is the number of reactions and compounds that were filtered. Yes, that's 899,000. So when I said combinatorial explosion, and that's with filtering, by the way. So like when I said combinatorial explosion, I really, really meant it. If we did not filter, this number after 3 generations would be far, far bigger than this. And then the number of generations, the starting compounds in my model was 1,364 with structures. It will only include structures, compounds with structures. So if you give me a model that has no structures in it, if you don't put structures in your SPML file and you upload that model in the KBase and you run it, it is not going to work very well. You have to give me structures. If you build a model in KBase, it automatically has structures because it's tied to our database. You've got compounds generated. So this is how many compounds are generated on top of the starting compounds. How many of the starting compounds were in model seed? How many of the generated compounds were in model seed? So it found 742. In 3 steps, it found 742 additional model seed compounds. So our rules are not perfect. If we had a more comprehensive rule set, which we are working on doing with our collaborators, this would be a much bigger number, I believe. But, these would be bigger numbers too. So it's a double-edged sword. How many peaks in our data set matched our compounds? So at the moment, I think for the FTICR data, this app, it's kind of weird, but at the moment, this app is the app you'd have to run to figure out how many of those FTICR peaks actually occur in your model. And the answer is 24 of 3,516 that were in this condition. So this was condition-specific FTICR. We ran all the peaks that had non-zero values in this specific condition, which was the ERP-A SW. It was 24. How many peaks matched generated compounds? 1,881. So more than half of the peaks, the cheminformatics found candidate pathways and candidate compounds for more than half the peaks, while the model contained only 24. So the cheminformatics is really expanding how much of the metabolomics can be, hypothetically explained by the model. Remember, we're in Wonderland, so it's only hypothetical. The predictions here are just that. They're predictions that need to be investigated further. But the nice thing is it gives you mechanistic hypotheses that lets you design experiments to actually do further investigation. How many peaks? Now, just as a crazy thing, this app, it doesn't need to do this for the app to do what it does, but it also tries to find all these peaks in the model seed. So if you take these 3,516 formula, I'll drill this point home. 3,516 formula that FTICR gave you after being annotated with formulator, and you compare it to the formula of every known molecule and KEGG, Metapsych, and other databases that are in the model seed, something like 30,000 molecules, known biochemical molecules, you get 768 hits. So that's dark matter. The 768 isn't the dark matter, but everything else. So there's a lot of, we only generated, 742 model seed compounds. So there's other model seed compounds that we didn't generate, but they are in the model seed. So the next step in this, although it's not a tool we have in KBase at the moment, is to integrate the chemistry from Pickaxe and the bottle seed into a model and try to activate and reach as many of these, you know, peaks as possible in a single flux solution. That's a tool we're working on now. But you would want to merge both the Pickaxe network and the model seed network so you could benefit from all of these findings. But these 768 are probably quite far, many of them are probably quite far from the compounds in your model. And they're formula matches. So again, formula matches, you want structure matches. Formula matches are, you know, really a good chance that they're not right. The starting compounds that hit the peak, there's 28. So there's 28 compounds in the model that hit 24 peaks. This is a very important thing to drill in. Multiple compounds will hit the same peak because multiple compounds will have the same formula. There's 15,000 generated compounds that hit 1,800 peaks. So this gets a lot where I mentioned previously, the model, you know, the cheminformatics is really good at once it finds one peak, one compound that hits a peak, it will find many more very, very quickly. So that's what it's doing. It's finding, you know, get the math here, 8 or 9 compounds, you know, per peak, and this based on this analysis. And then the model seed compounds with peaks, 2,071. So if we go up to generations, the generations tab shows you how many compounds you had after every generation. So you started with 1364. We ran 2 rule sets with a limit of 3,000. So it's keeping 6,000. If it keeps 8,000, it means like your filtering criteria was maxed out. So there's no random sampling anymore at this stage. It's keeping everything because it's hitting that many peaks. So it kept 6,000, then 8,000, then 10,000. It filtered 26,000 in the first generation, 291,000 the second generation, and 415,000 the third generation. So this is an interesting phenomenon because this is scaling faster than the number of input molecules. And this is because of chemical diversity. You're inputting a broader chemical diversity with each generation, and this is leading to even more chemistry being generated. So that's the other issue of combinatorial explosion, is it's not just exponential increase, it's like exponential of an exponential. But the nice thing is this tool, because it keeps all the peak hits, or at least a representative set of them, lets you very quickly generate chemistry to reach, you know, over half the peaks you found. Now, I mean, these may not be right, but at least you have candidate pathways to look at, and then you can use experiments and correlation and other things to try and find if those try to see if those predicted pathways and compound structures are right. We have rules. So this just lists all the rules and how many reactions came from each rule. So you can see like the most aggressive rule. So 2.1.1, the enzymatic rule 2.1.1, which is just the EC number 2.1.1. So if you look at reactions with the EC number 2.1.1, you'll see what this rule, approximately what this rule does. And there should be a picture of the rule, but we've temporarily removed those. And then the peaks. So this is all the peaks that you have and what compounds were found for each peak and whether or not they were in the original model and Pickaxe or wherever. And the compounds and reactions, currently it's only showing the first 100. I'm hoping to fix this, but it only shows the first 100. But if you want to see the full set of chemical reactions and compounds predicted, the way to do that is if we just search for, just click on the reaction here. Like, I'll do this one. I'm not connected to my narrative, so I'll have to reload. But if you click on the model and the data panel, you'll see what that reaction, what that full model looks like, and including, you'll actually be able to see all the chemical structures that were predicted. So you can see what those compounds actually look like. So that's the workflow and KBase. Of course, I've restarted this thing, so I can't. I'll have to wait for it to load. While we're waiting for this, I can take questions. That's that's what I have at the moment.
So thank you, Chris. That was fascinating. There's been, there's not really been any questions into the Zoom chat. I know there's been an ongoing discussion over in Discord. One question that just came up was, does Pickaxe filter out thermodynamically improbable products and formulas, or do the enzymatic rules already account for this?
The enzymatic rules will account for this to some extent, but only very limited. There's some crazy structures that'll definitely come out of this, and so we are looking at like my collaborators, the Broadbelt Lab and the TIO Lab at Northwestern. They're really looking at this, adding our group contribution based cheminformatics methods, integrating that into Pickaxe so we can run those. And if you get a group that the group contribution method just doesn't even recognize, well, you could potentially just throw those out. So that would, we will add those to Pickaxe as that those methods mature, and those will show up as new checkboxes in this interface. So you'll see new checkboxes with the thermodynamics. Like there might be a threshold for the predicted delta G value. So what we could filter everything below that threshold, everything with a delta G above that threshold, a really large delta G for the reaction.
Okay, great. Yeah, so when you have a chance, if you want to take a look over there, I know Jonica's been busy answering a lot of the questions already over in Discord. One thing I would be interested in, Chris, if you would kind of give us your, I don't know, I'm sort of interested to hear about your perspectives on how this field has advanced over the last, say, 10 years, or, where some of I mean, this is certainly an exciting advance because you're able to now bring metabolic information together with metagenomic information to really build better models. So, I mean, my perspective is I remember, and maybe my memory is bad, but it seemed to me about 10 years ago or so, when I was just learning about genome scale modeling and FBA, I remember going to a SciDAC meeting and talking to you at a poster and being really excited because you were starting to have tools for automatically annotating genomes, and I thought that was amazing at the time. And maybe my timing is off on that, but KBase at the time wasn't even a thing, and so how fast the tools have developed and where we're at today, the things you're showing. If you could just kind of give our listeners your perspectives on how fast that has happened and what some of the potential is going forward, that would be really interesting to hear.
Yeah. So I think this field is really taking off. I might actually have an open narrative over here. I have another set of brown tabs. Yeah, here we go. Because this one's not connected either, so I won't be able to. So I think the metabolomics and the genomics combined are really, the metabolomics technology is really exploding right now. And I think we're in a, in 1990, which is 30 years ago now, depressingly. There was this exciting revolution in sequencing, and then, with the first genomes emerging, and then in the 2000s, next gen sequencing coming along and like blowing things up again. And there was this golden age of discovery where we went from, I remember we had hundreds of genomes. And I was like, we built the model seed, and we built like 130 models with the model seed, and it was a large fraction of the genomes that existed in the day. And then we're like, let's build 3,000 models, and that was a large fraction. And now there's, over 400,000 genomes, and that's not counting the mags. I think with metabolomics, we're getting there. We're about to enter a new age of metabolomics, because a combination of machine learning, being able to annotate metabolomics data better, and the combination of improved instrumentation. So I talked about, like, well, they're working on adding MSMS to FTICR, and the non-FTICR machines, Orbitrap, and whatnot, are getting better too. And the separations technology is getting better. And by getting better, I mean, like, currently retention time gives you some very limited information. It basically tells you, like, this peak is different from this other peak because it has a different retention time. That's kind of what it tells you at the moment. But new technologies that are coming along for separation that are not GC and LC, liquid chromatography or gas chromatography, those new technologies might give you a lot more information from the retention time. They might be able to separate stereoisomers. This is ion dispersion and new techniques. And the columns are getting a lot longer because the instruments are getting smaller. So all of this is coming together at the same time with machine learning to make a golden age of metabolomics. And if you can see the chemistry, then the tools I've just shown you can map the chemistry together. And it goes from currently you have this, unknown proteins, which is all you have. You have biologists working in labs to discover painstakingly, one at a time, individual chemical reactions. And, eventually, a year later, those new reactions get put into databases. And then a year later, all the annotating algorithms get updated, and then those new reactions might actually show up in a model. And I'm being very generous to say that'll happen in 2 years. But now imagine that you discover all the chemistry, and it becomes a big connect the dots with computational tools that integrate genomics data. And the computational tools to do that connect the dots, I would argue, the Pickaxe tool combined with all the advanced annotators combined with RNA-seq data, they're getting there. So we'll be able to connect the chemistry of an environment overnight, and then do high throughput. There's a high throughput instrumentation, so high throughput confirmation of predicted functions and predicted reactions on robotic systems, using droplets that can float magically through the air. That's where we're at today. So we're in a golden age of biochemical, biological, enzymatic discovery, which is going to fuel a golden age of synthetic biology. So yeah, I think it's an exciting time.
Mm-hmm. Thanks for that perspective, Chris. I think, it's important for the summer school participants to, understand that the things that they're seeing this week really are, on the, I guess, the bleeding edge of, the most advanced work that's being done today. So, if people are having a difficult time understanding and following, understand that, the people who are doing this work are really in the stages of, just developing this material. So this is an opportunity for, as you're learning about these things, to get engaged with and to get involved with things that are really exciting and moving forward at a rapid pace. So thanks again, Chris and Jonica, for sharing your expertise with us, and Hyun and Mark as well. This morning, it was a really fantastic session, and I think seeing these things coming alive in KBase and being accessible to a lot of researchers is making it even exponentially more powerful for everybody. So